Inherited breast cancer. So this is, we're going to be talking about a specific type of cancer. And breast cancer is very common. So the objectives for this lecture include comparing the risk of breast cancer in the general population to those that have a presence of a BRCA1 or 2 mutation, identifying other cancers seen in families with inherited breast cancer, recognize the risk for breast cancer in males with a BRCA1 or 2 mutation, and finally identify additional genetic causes seen in inherited cancer. So breast cancer is common. Approximately 12 to 13% lifetime risk to develop breast cancer. 1 in 9. Some things I'm seeing says 1 in 8. They just keep increasing it. Males have approximately a 0.1% chance to develop breast cancer in their lifetime. So males don't have as much breast tissue, so they have a lower risk of developing breast cancer. Most breast cancers are not hereditary. Approximately 3 to 5% of all breast cancers are due to a mutation of the BRCA1 or BRCA2 gene. And more genes are being linked to hereditary breast cancer, but these are the main players. These are the ones you really need to understand. So here's a family history of a family with breast cancer. And this woman is the person that came in, and she had breast cancer at age 45. Her mother died of ovarian cancer at 65. Her mother's father had prostate cancer at 55. She has a sister who has a son and a brother who has 2 daughters that developed breast cancer, one at age 54 and the other at age 35. So on the woman's father's side of the family, there's really no cancers going on. So the concern here is that we're seeing ovarian cancer, early breast cancer, prostate cancer in a male at a relatively young age. So those are signs of an inherited breast cancer. So early onset of cancer, bilateral breast cancer, ovarian cancer, including fallopian tube cancer. Some believe that the BRCA1 and 2, it usually starts in the tube, but again, by the time it's diagnosed, it's in the ovary. Pancreatic cancer, multiple family members with breast cancer, and male breast cancer. Again, male breast cancer is super rare. When we see it, we're worried. The gene for BRCA1 is located on the long arm of chromosome 17. And this gene provides instructions for making a protein that acts as a tumor suppressor. A tumor suppressor protein helps to prevent cells from growing and dividing too rapidly or in an uncontrolled way. The BRCA1 protein is involved in repairing damage to DNA, and when the gene does not function correctly, this DNA damage accumulates every time the cell divides. The BRCA2 gene, and the way they got the names, they're starting to name things better than they used to. The BRCA1 gene is the first breast cancer gene that they found, and the BRCA2 is the second one. This gene works with other genes and is involved in repairing DNA at the G2M stage of the cell cycle. And when non-functioning correctly, damaged DNA accumulates, results in increased risk of breast, ovarian, prostate, pancreatic, and melanoma. So let's, again, go back to our trusty cell cycle, and it works at G2. The BRCA1 and the BRCA2 work at G2, and it prevents the cell from undergoing mitosis if there's damage. When an individual is born with one non-functional copy of this gene, with the Knudsen two-head hypothesis, the second mutation is also needed before there's problems. So you don't develop cancer with just one copy, you need both. So 89% of females born with a non-functional copy of either BRCA1 or 2 gene will develop breast cancer. The other 11%, they were born with that one copy that didn't work, but they were lucky that in no places in their breast did that second copy get lost. There are actually 3 common mutations, 2 in the BRCA1 and 1 in the BRCA2 in the Ashkenazi Jewish population, although there are hundreds of changes in this gene. And we don't do common gene mutations much anymore, but sometimes people just look for common genes, but if you're not of Ashkenazi Jewish descent, then you're probably not being tested. Anyway, hereditary breast and ovarian cancer, as I said, is a germline in every cell in our body change of the BRCA1 or 2 gene. It's inherited in autosomal dominant. So let's look. If you have a mutation of a BRCA1 or 2 gene, the risk of developing breast cancer is 89%, not 100. 11% people get lucky. Whereas in the general population, a woman's risk is 12 to 13%. So if there's a family history of a BRCA1 or 2 mutation and they test negative, they drop down from 89% to still 12 to 13%. There's no get out of cancer free card. You still need to get your mammograms. And there are a number of families with the pedigrees that some people develop breast cancer, but they got the sporadic kind. So you still are at risk of this. A second breast cancer. If you have a mutation, there's a 64% chance that you will have a second breast cancer. In the general population, if you develop breast cancer, there's an 11% risk. Male breast cancer, again, very, very uncommon in the general population, but 6 to 8% if a male carries a BRCA1 or 2 mutation. Now, I might like to point out that 6 to 8% is still far lower than just a woman in the general population, but you know, it's a much higher risk than 0.1%. Ovarian cancer, about 1.5%. If you have a BRCA1 or 2, it's 44%. And pancreatic, 7 to 8%, with about 1% in the general population. Now, these numbers change a lot. Not as much these, but down here, do not memorize these numbers. Just know that male breast cancer is super rare, about 1%. These are not the things to memorize. It's just to get a perspective. So the question people sometimes say is, why get a genetic test to find out if I'm going to develop breast cancer? Like, why would I want to do that? And 15 years ago, it's a hard argument, you know, really expensive. What are you going to do if you come from a strong family history of breast or ovarian cancer? Why would I do this? The answer today is management, because there's actually a lot of risk, things that you could do to decrease the risk of breast or ovarian cancer. So if we have a woman or even a man, but we're primarily talking about women, with a BRCA1 or 2 mutation, if a woman has a BRCA1 or 2 mutation and she stays on oral contraception, she can decrease her risk of ovarian cancer by 50%. Just by staying on birth control. Tamoxifen can decrease the risk of breast cancer by 49%. Having a mastectomy can decrease your risk of breast cancer by 90%. And actually having your ovaries removed decreases your risk of ovarian cancer by 90%, but also decreases your risk of breast cancer by 68%. So stay on birth control till you have your kids, and when you're done, have surgery. And breast cancer testing really took an upbeat with the Angelina effect. Angelina Jolie actually came forward to the whole country, which I thought was incredibly brave, to explain why she had this risk-reducing surgery. And it doesn't take away 100% risk of breast cancer, but it does decrease it significantly. If no surgery, then we need to start doing surveillance. And this usually involves breast MRI as well as mammograms starting at age 25. Recommended risk-reducing bilateral salpingo-oophorectomy. And genetic testing for these genes are associated with breast cancer increased, like I said, after Angelina Jolie came out and explained why she had. So stay on birth control till you have your kids. You know, come off of it, have your kids, and have breast-reducing or, cancer-reducing surgeries. Additional single genes associated with breast cancer. We talked about Lee Fermini in the last presentation, the worst gene ever. It controls many stages of the cell cycle, so the risk of, if you have a germline mutation in Lee Fermini, there's an 80% chance of breast cancer, 22% of brain cancer, sarcoma. You know, when you see the word sarcoma, you should think about Lee Fermini. Mostly they're sporadic, but you're always worried about that. Colorectal cancer, pancreatic cancer, bone cancer, leukemia. and this stands for adrenal cortical cancer. So here, again, is our pedigree of the Lee Fermini, that there's cancer everywhere. Breast cancer at 30. Her dad died of osteosarcoma at 25. He had a brother that died of leukemia, which usually is not inherited, but, you know, colorectal cancer at 50, breast cancer at 39. And then on this side of the family, somebody that died of lung cancer at 65, and he was a smoker. So we don't worry about these people too much. But this is a pedigree of a Lee Fermini. Other genes, CDH1, this is associated with hereditary diffuse gastric cancer, but you can also have an increased risk of lobular breast cancer and diffuse gastric cancer. P10, PTEN, there's certain, one of the things we realize is some genes, some changes in the gene cause one thing and some cause another. Cowden syndrome is an inherited disorder, but there's an increased risk of breast, follicular thyroid, and colorectal, as well as renal and endometrial. They have a big head. They can have thyroid goiters, GI polyps, learning problems, and skin lesions. STK11, Peutz-Jeghers syndrome. There's an increased risk of breast and colon cancer, and there can be hyperpigmentation of the lips. So let's just look at the CDH1. It codes for this gene called calhierin, and it's involved in cell adhesion. Impaired cell adhesion increases the likelihood that cancer cells will not come together and they'll invade into the stomach. 20 to 40% of diffuse cancers are caused by a mutation of CDH1, and they're also associated with, as I said, lobular breast cancer. Cowden syndrome is caused by the P10 gene mutation located on chromosome 10. Breast, thyroid, colon, and uterine cancers usually start developing between 30 and 50. And another sign of Cowden syndrome is having a really big head. Other features include having this trichomania, breast cancer, endometrial cancer, thyroid, and they can have hematomas, intestinal polyps, fibrocystic breast disease, and lipomas. Peutz-Jeghers, they love to ask about this, so if you ever see this picture, that's why I'm showing it to you. There's these freckling on the lips. It's a dominant and STK11. But in addition to those freckling on the lips, there's a 32 to 54% risk of breast cancer, colon cancer about 39%. They have these hematomatous polyps. They can also increase the risk of bowel obstruction, stomach cancer 29%, and ovarian. So understanding family history patterns, many of the inherited disorders we have discussed have overlap. The BRCA1 and 2 are the most common, but P10, you might have breast or endometrial colon, Peutz-Jeghers, breast, ovarian, colon, stomach, freckling of the lips. TP53, like literally any kind of cancer, sarcomas, leukemias, brain tumors, breast, anything, but they happen really, really early. And so in this family, the individual is worried because her mom had breast cancer. She had breast cancer at 44 and she's now 59. She has a brother who's 56, alive and healthy, but a sister who died of breast cancer at 50, but she developed it at 48. And her maternal grandfather died of prostate cancer at 62. On the dad's side, no real prostate at 61. He's 62 now. So we're concerned about a maternal history of cancer. And this is a pattern we're seeing, with prostate, that's not that exciting, but we have, 2 early breast cancers in 2 different sisters. And so we're also, the other concern is individuals of Ashkenazi Jewish descent. Whenever we do a family history, we always ask people's ethnic background because there's certain genes that might be more common in certain populations. It's just part of what we do in genetics. Every ethnic group is at risk of something. So it just helps us when we're doing risk assessments. But this girl would not be the best person to test. Her mom's still alive. if her mom carries a BRCA1 or 2 mutation, each of her children is at a 50% risk of inheriting it. If I test her and she's negative, is she negative because she didn't inherit it, or is she negative because there's something else, a different gene in the family? So if there's a living person, even if it's a cousin, somebody that's a better person to test, I always want the person with the cancer or the disorder to test, because then a negative test can give us some reassurance. So if we find that she has a BRCA1 mutation, we can test the daughter for that same, and her 2 brothers for that same change. If we don't find a genetic change, then genetic testing is really not going to change her risk. So we really want to always test the person in the family that, you know, has the highest risk of being positive, so that we have something informative. So in conclusion, inherited genes can increase the risk of cancer. Most tumor suppressor genes are inherited and autosomal dominant, as I said in the previous lecture. 1 copy of the gene is non-functional from conception, but the 2nd copy is lost as a somatic change in a group of cells. Tumor cells usually have 2 non-functional or abnormal copies of the gene. Germline usually only have 1. This is becoming more and more important as they're doing more and more somatic testing on tumors. So if you did somatic testing on a breast cancer and you found a BRCA1 or 2 mutation, that does not mean that that individual has inherited. They could have lost both of their copies somatically, but sometimes they see things, some of the tissues have 2, some have 1, then that makes them to want to possibly test the person for an inherited cancer. So that concludes this presentation.